Today we will be starting on the different processes of biotechnology. One of the most important processes of biotechnology is what happens after we identify the particular sort of a gene of interest, and we have already amplified the gene of interest using a polymerase chain reaction. Once we have amplified the gene of interest or made multiple copies of the gene of interest, then normally if you take cells, if you take individual cells, a gene of interest develops as a sort of a recombinant protein on a small scale in a lab. If ever we want to take your gene of interest and develop it in a test tube, or we want to develop it in a sort of a petri dish, if in a petri dish we have your gene of interest and we want to make multiple copies of the gene of interest in order to produce the sort of a chemical. In the gene of, we have the gene of interest which is present in a sort of a petri dish, and it has got a medium, and your desired product, which is the sort of a chemical, which is formed from the recombinant gene, can easily be produced in the lab. But however, when you produce it in the lab, it is of a small scale. Now, if we go back to the definition of biotechnology, biotechnology primarily aims at development of the gene product or the amplification of the desired gene to produce industrially important compounds which have primarily applications in agriculture, medicine and environment. Thereby, it is very important that we exactly know what is the way of how do you amplify your gene of interest and go in for large scale production. Now, for large scale production, the production from the recombinant DNA can be obtained on a large scale by using certain large vessels which are termed as bioreactors. So, the goal of bioreactors is for large scale production, for large scale industrial production. If you take your sort of a recombinant DNA which has already been amplified, we want to go for large scale production of antibiotics, enzymes, vaccines, drugs, pharmas and other sort of natural bodies. We go in for a sort of a bioreactor. Now, what exactly are bioreactors? Bioreactors are large vessels. They are large vessels which could be about maybe the size of an inverted sort of a train engine. They are large vessels and they can hold up a volume of 100 to 1000 liters. So, in simple, if you take a bioreactor, will have a curved surface, it will have an inlet valve, it will have an outlet valve. It will have an inlet and an outlet valve, and your specific gene product are agitated by microbes under controlled conditions of temperature and pressure. Under controlled conditions of temperature and pressure, you will exactly take cells or specific gene products which have been engineered by microbes and placed in a bioreactor. Now, a bioreactor what does the bioreactor provide? The bioreactor primarily provides optimum conditions. It provides the optimum conditions of temperature and pressure, and it also provides a facility for the inlet of your sort of a recombinant DNA molecule. Thereby, here the raw materials which are biologically produced, the raw materials which are biologically produced from recombinant DNA technology are fed into the sort of a bioreactor. They are fed into the bioreactor. Now, when you take a typical bioreactor, it provides the optimum conditions for obtaining the desired product. We have the desired product which you get here. We get the optimum, not the maximum, not the minimum, but the most efficient quantity. The efficient quantity is termed as optimum, and it provides optimum growth conditions. What does the bioreactor have? It can provide optimum growth conditions such as temperature, such as pH. It provides temperature and pH, and also it provides the different sort of a substrates which are necessary for the growth of the particular sort of an organism. It may also include addition of salts and whatever be so. So, it has an inlet. It has an inlet for a large volume of the culture medium. When you take the culture medium is fed through the sort of an inlet valve, and it also has an outlet. It also has an outlet from which the spent media is thrown out. The spent media, the used media which is important for the culturing is taken out, or the waste is also dispensed. At the same time, you also have your desired product coming out of a sort of a bioreactor. And among the bioreactors, we have got different types. The most common of the bioreactors is termed as the stirring type bioreactor.